Hello students, welcome to Zoology Classroom. In today's lecture, we are going to discuss about biotechnology, its scope, importance and applications. This topic comes under MSc Zoology, 4th semester, Animal Biotechnology paper, Unit 1, topic 1.1. So first of all, let us know what is biotechnology. Biotechnology is the manipulation of living organisms or parts of the living organisms to make products useful to humans. And this biotechnology deals with the manipulation of genes of organisms to alter the behavior of animals, characteristics, or value of the animals. Cell and gene technology. Gene technology means rDNA technology is used to produce new characteristics in plants or animals. So simply we can say that biotechnology is nothing but using the technology in animals or plants or in living organisms for the benefit of mankind. So various definitions have been given for this biotechnology. Some of the definitions are given in this slide. So let us see a few of them. If you want, you can take screenshot and you can read them at your leisure. So biotechnology is a collective noun for the application of biological organisms or biological systems or biological processes to manufacturing and service industries. So the integrated use of biochemistry, microbiology, and engineering sciences in order to achieve technological application capabilities of microorganism, cultured tissue cells, and parts thereof. So biotechnology is a technology using biological phenomena for copying and manufacturing various kinds of useful subjects. So these are the few definitions given for this biotechnology. So simply we can conclude that biotechnology, in this term biotechnology, we have two parts, bio and technology. So technology is related to using the knowledge of science for the benefit of mankind. So technology is a branch of science in which scientific principles will be utilized for the benefit of mankind. And biology is a branch of science. So here in this technology, we are using the rules, principles, knowledge of biology for the benefit of mankind. And in technology, invariably, we need to use certain instruments, certain machinery, or certain tools. So, and all these tools again will be made by utilizing the knowledge of physics and some other branches of science. So in this way, biotechnology uses the knowledge of biology, the knowledge of physics, the knowledge of chemistry, and knowledge of other branches. So in this biotechnology, we are utilizing the biological organisms, be it animals, be it plants, or be it microorganisms, and we are exploiting them. We are exploiting the characteristics of biological organisms for our benefit. So that is the scope, that is the area of biotechnology. And let us see timeline of biotechnology. Since ages, man using this biotechnology, 8000 BC, okay, human beings have been domesticated plants and animals almost 10,000 years ago. 8000 BC means almost 10,000 years ago only humans have been domesticated plants and animals. So now, then only we have started using the characteristics of plants and animals for our benefit. So that we can say that the beginning of biotechnology was around 10,000 years ago. 4000 BC, that means around 6,000 years ago, Egyptians have the knowledge of making wine from some plant products. So this is the uh, domesticating is a simple way of using animals, but making the wine is not a simple task. So this requires a lot of knowledge. So 6,000 years ago only Egyptians have mastered in that art of wine making. And 4,000 years ago, Egyptians again, they used yeast to make bread. And 4,000 years ago, Egyptians and Sumerians learned brewing and cheese making. And 2,500 years ago, mildly soybean curds used to treat boils. So soybean, mold soybean and curd, these things were used to treat the boils or we can say source of the patients. So to treat the boils or source of the patients, these things have been utilized around uh, 2,500 years ago. And 2,300 years ago, Greek people developed grafting techniques. So grafting techniques, still we use grafting in plants. So this grafting technique was first developed by Greeks around 2,300 years ago. And in the year 100, that means 1900 years ago, chrysanthemums were powdered and used in certain operations or in certain activities. And in year 1663, Robert Hooke discovered the cell. And so far, what happened till this year, 1663, was traditional biotechnology. In this thing, we need not to apply the knowledge of genetics or any other modern biological knowledge. But now biotechnology is having a lot of scope, and biotechnology now using the knowledge of modern biology, that means genetic science, and molecular cell biology, all these fields of all this knowledge of all these fields is being utilized in modern biotechnology. So modern biotechnology is quite different from the traditional biotechnology. In traditional biotechnology, we just use the microorganisms to make curd or to make cheese and to make breads and to uh, use the organisms for their products. So all these are simple ways of using organisms for our benefit that makes the traditional biotechnology. But modern biotechnology is unimaginable, and the foundations for modern biotechnology were laid with the discovery of cell by Robert Hooke in 1663. And in 1675, Anton van Leeuwenhoek discovered some protozoans and bacteria under microscope. And in 1997, Edward Jenner created cowpox vaccine. This is also another turning point in the timeline of biotechnology. So, and in 1802, biology branch was created by persons, scientists. And in 1830, proteins were discovered. In 1855, Escherichia coli was discovered by Theodore Escherick. And 1857, fermentation and germ theories fermentation was discovered and germ theory was proposed by Louis Pasteur. And 1859, uh, Charles Darwin published the theory of evolution by natural selection. In 1861, Louis Pasteur developed pasteurization technique. And in 1865, Gregor Mendel laid foundations for genetics branch of science. 1888, Henrik Wilhelm Gottfried Waldeyer discovered the chromosomes, or simply Waldeyer discovered the chromosomes. In 1915, bacteriophages were discovered. 1919, biotechnology term was used by Karoli Erecki. So this is the person who used biotechnology word for the first time in 1919, Karoli Erecki. And in 1922, Dr. Frederick Banting and Charles Best. Banting and Best discovered insulin enzyme. And in 1927, Hermann Muller uh, discovered that radiation causes defects in microsomes. And in 1928, Alexander Fleming discovered antibiotic penicillin. In 1944, Avery, McLeod, and McCarthy proved that DNA carries the genetic information. This is also another turning point. And in 1953, the biggest milestone in the timeline of biotechnology is discovery of structure of DNA by Watson Crick. and Wilkinson's X-ray diffraction method used for this discovery. In 1966, a genetic code was cracked by Nirenberg and Robert Hole and Hargobind Khorana. And in 1971, for the first time, a artificial gene was synthesized in 1971. And in 1973, Stanley Cohen and Herbert Boyer perfected genetic engineering techniques. So rDNA technology was discovered by these two people, Stanley Cohen and Herbert Boyer. And in 1975, again, George Kohler and Caesar Milstein developed the technology to produce monoclonal antibodies. 1982, first FDA approved human insulin was produced. So in America, FDA is the authority to give permits for the newly discovered drugs. So that institution gave permission for human insulin production. And in 1981, first transgenic animal, the rat was produced. So this is the first transgenic animal. In 1983, PCR technique, polymerase chain reaction technique was discovered by Kerry Mullis. In 1986, first recombinant vaccine for hepatitis B disease was made by the scientists. In 1986 alone, uh, first anti-cancer drug, interferon, was produced. In 1987, virus-resistant tomatoes, first genetically modified plant, was produced. In 1994, first GMA product was sold in the USA, that is tomato. And in 1997, first cloned animal, Dolly, was made or produced. In 1998, human embryonic stem cell lines are established. 1999, Human Genome Project was launched. 2002, the Human Genome Project discoveries were published. In 2003, SARS virus was sequenced, severe acute respiratory syndrome. In 2004, first clone pet was produced. In 2006, recombinant vaccine for human papilloma virus was produced. In 2010, malaria-resistant mosquitoes were produced. So these are the milestones in the timeline of biotechnology. Now let us see about the scope of biotechnology. So biotechnology is a multidisciplinary branch of science. This has emerged as a demanding industry during in the recent past. So as I have already told, biotechnology requires the knowledge of biology. In this biology, again, knowledge of genetics, microbiology, zoology, botany, cell biology, molecular biology, like that, several branches of biology, and knowledge of physics, chemistry, knowledge of computer sciences, knowledge of bioinformatics, knowledge of statistics. All these branches of science are required in this biotechnology. And this biotechnology has been developing a lot in this recent years. Why? Because our requirements are as such. Besides being a branch of advanced biological sciences, it attracted many multinational companies, including those are concerned with. So many multinational companies are interested in doing business in biotechnology branch. Why? Because these multinational companies are interested in producing pharmaceutical products for many diseases, and they are also producing many antibiotics, vaccines, and life-saving drugs, and gene therapy products are all being produced by these multinational companies. And this industry requires the knowledge of biotechnology. And these companies, again, they are also improving the clinical testing methods and diagnostic tools. So recently we can see that for this COVID-19 pandemic, rapid antigen test, earlier it was not there for identifying the COVID-19 virus. So these rapid antigen tests, RT-PCR tests, are developed for identifying COVID-19 vaccine. So for each new challenge, a new testing methods and new diagnostic tools have to be developed. So all these companies are doing business in this field. So RT-PCR test and antigen test, the basic principles are already there, but identifying the particular organism. So how to identify the particular organism? For that thing, they require a little more research and a little more work in that way or in that angle. So they are doing work in this field, and they are developing new diagnostic tools and new testing methods. And they are also interested in producing novel varieties of crop plants and animals, like genetically modified plants, genetically modified animals are being produced by these multinational companies by using this biotechnology knowledge. And production of wide range of food products and fertilizers, pesticides, and beverages are also being produced by utilizing the knowledge of biotechnology. So, by these multinational companies. And waste treatment methods, bioremediation methods, and energy production in energy production fields are also biotechnology is being utilized. And production of reagents, including enzymes, DNA, RNA, etc., are also being produced. And this biotechnology has also enhanced the sale of many biological products in the world market by improving their varieties and by increasing the production amount or production quantity. And the improved varieties include alcoholic beverages, antibiotics, enzymes like penicillin, tetracyclines, etc., amino acids, vaccines, steroids, vitamins, insulin, human growth hormones, microbial pesticides, citric acid, improved varieties of seeds, etc. So this is about scope of biotechnology. Now let us come to the applications and importance of biotechnology. So biotechnology knowledge is being utilized in microbial biotechnology branch. So new branches have been formed by applying the knowledge of biotechnology. So microbial biotechnology, agricultural biotechnology, aquatic biotechnology, bioremediation, animal biotechnology, medical biotechnology, and forensic biotechnology. So coming to microbial biotechnology. So microbial biotechnology is nothing but using the microbial organisms, knowledge of microbial organisms in this biotechnology field. So here in this microbial biotechnology, we use the knowledge learned by the microbiology field in this biotechnology for the benefit of mankind. And in this microorganisms, usually bacteria and viruses are also being utilized in this microbial biotechnology field. Usually yeast and many bacteria are being used as a tools in this microbial biotechnology. And all these microorganisms are being used for creating better enzymes and for creating more efficient decontamination processes for industrial waste product removal. And they're also used to clone and produce large amounts of important proteins used in medicine. For example, human insulin is being utilized by these microorganisms only, this bacteria only, and many other drugs like interferons, uh, uh, like that, many products, many enzymes we use as a medicine or for curing diseases are being utilized by the knowledge of this biotechnology and microbiology. So this makes the microbial biotechnology, and it has a lot of scope in coming years. And another one is agricultural biotechnology. In this, we use the knowledge of agriculture and we apply them into biotechnology so that we can get benefits from this agricultural biotechnology. So the purpose of this agricultural biotechnology is used to produce plants that are environmentally friendly and that yield a lot of products per acre. So genetically engineered plants, production of genetically engineering plants is the core area in this agricultural biotechnology. And we use this genetical engineering technology in this agricultural biotechnology to produce disease resistant plants and pest resistant plants. And we also use this knowledge to produce foods with higher proteins or vitamin content. And in some cases, we also produce plants which have some drugs in their products, like in their seeds or in their fruits or in their leaves, like that. So by eating those drugs, by eating those plant products, be it seeds or be it fruits or be it plants or be it stems, any other part of the plant. So by eating the plant parts, we can get the drug. So in coming years, in future, we may see a day that if we have a disease, we need not to take medicine. Instead, we can take certain type of plant products so that we can get the medicine in that plant product itself. So in that way, many researchers are being going on, and they are going to be available for the common people in coming years. And these better plants, ultimately what they will do, they will reduce the production cost and they will help the growing needs of the food of the world population. And third one is animal biotechnology. So in this, we use the knowledge of zoology, and we use animals in this animal biotechnology, and we will try to get more benefits from these animals by applying biotechnology knowledge. And we use animals for the production of antibodies, and transgenic animals will be produced. And in these transgenic animals, we will get antibodies, we will get enzymes, or we will get many useful products from those animals. And animals are also being used as important models in basic research activities. Uh, we know that before the COVID vaccine is prepared and made available for public, the research trials were conducted on rats. So animals will be utilized in doing these type of researches. So, and gene knockout experiments also will be conducted in animals before they are carried out in human beings. And design and testing of drugs and genetic therapies are also will be conducted in these animals before they are made available for public. And animal cloning also being conducted with the knowledge of this animal biotechnology. And next one is forensic biotechnology. So in this forensic biotechnology, DNA fingerprinting is the major technique being used. So in this, the knowledge of forensic biotechnology will be used in solving some crime cases. So if we are suspecting a person, whether he committed a crime or not, so that thing can be clarified by using the forensic biotechnology. And paternity issues are also will be solved by this DNA fingerprinting technology. And identification of human remains, and endangered species, identification of endangered species, and tracking and confirmation of the spread of the disease. So all these things, they require the knowledge of forensic biotechnology. So here, coming to this last point, trafficking, tracking and confirmation of the spread of the disease. So here in recent times are also we are hearing the news of this where this COVID-19 vaccine being produced, or from where it has spread to the world. So for this researcher also, some scientists from World Health Organization have gone to China. So what they will do, they will use the knowledge of this DNA fingerprinting and forensic biotechnology only. So to confirm the area from where the COVID-19 vaccine was first spread. So all these things are also will be using the knowledge of forensic biotechnology. That means DNA fingerprinting. And next one, very important one, bioremediation. So with the modern industrial revolution, we know that we are, apart from getting benefits of this industrial revolution, we are also facing a lot of problems. That is a very, very important problem is environmental pollution. So, answer for this problem, the answer for environmental pollution is bioremediation. So in this, we use the knowledge of biotechnology. Biological organisms will be utilized to degrade the pollutants. So, and these bioremediation methods are natural, and we will produce by the knowledge of, we will produce certain substances, materials, by the knowledge of this biotechnology. And these products, they are environmentally friendly, and they do not cause any damage to the environment. Apart from that, they also help us to clean the environment, or they also help us to degrade the environmental pollutants. So in this way, bioremediation has a lot of scope in coming years. It is going to solve our environmental pollution issues. So, for example, stimulated growth of bacteria that degrade components in the crude oil. So in 1989, Exxon Valdez oil spill in Alaska region. Alaska is a state in America. So here in this Alaska region, oil spill issue was generated, or oil spill issue took place. So while transporting the crude oil through the sea, this type of accidents occur, and where the crude oil is spread on the seawater. So that is a major issue. The life that lives in the sea waters, usually many animals will die because of this oil spill. So to solve this issue, some bacteria were utilized. So these bacteria, they degraded these oil spills, and they made clear the waters of the Alaska Sea. So for the first time in 1989, this bioremediation technique was used to solve this oil spill problem that took in Alaska area, Alaska region in 1989. And in 2010 also, deep water horizon spill issue was also solved by this bioremediation technique itself. And in this bioremediation, some nutrients will be added to stimulate the growth of the bacteria, so that the oil spills or the pollutants which are present in the soil or water bodies, all those things can be cleared by the bacteria. So Alcanivorax burkimensis is a bacteria. So this bacteria is being utilized in treating these oil spills. And aquatic biotechnology. So aquatic biotechnology is also very helpful to us. It helps us to meet the growing demands of food requirements. So in this aquatic biotechnology, aquaculture knowledge, genetic engineering or DNA technology knowledge, and bio prospecting methods will be utilized. So in aquaculture, usually we will grow different types of animals that live in water, usually two types, finfish or shellfish. And these things, these finfish and shellfish, they will be controlled by using the biotechnology knowledge, so that they grow big in size. So here in this thing, salmon fish, this is the normal salmon fish, and this is the salmon fish developed by our DNA technology. So this is genetically modified salmon. So we can see the difference in size of these animals. So by using this biotechnology in these aquatic animals, we can increase the production, so that this increased products, the increased quantity of these animals meets our food demands. And 50% of all fish consumed by humans worldwide. So in these fishes, human growth hormones were incorporated. By doing that, these genetically modified salmon fish were produced. In genetic engineering, in this genetic engineering, disease resistant strains of oysters are being produced, and vaccines against viruses that infect salmon and other finfish. So and transgenic salmon that overproduce the growth hormone, this we have already discussed. So not only we use this biotechnology in this aquaculture, just for not only for increasing the production, but also these animals, as they are also animals, they are also prone to lot of diseases. We can use the knowledge of biotechnology to make them disease resistant, and we can make many vaccines or many medical drugs to help them in defeating these diseases. And bioprospecting, so rich and valuable sources of new genes, proteins, and metabolic processes with important applications for human benefits can be developed and can be separated from this genome of these organisms. So in some animals, say some very new genes may be present. If we remove that gene and utilize in some other animals, we can get benefits from that animal. So in this thing, in this bioprospecting, the scientists will be studying the effects of the genes, and they will try to separate those genes, and they will try to incorporate those genes in some other animals, so that we can get more benefits from such genetically modified organisms. And marine plankton and snails found to be rich sources of anti-tumor and anti-cancer drugs. So here we can see in some marine plankton and marine snails, some genes were discovered whose products are having anti-tumor or anti-cancer characteristics. So if we are able to separate those genes, and if we are able to incorporate those genes in animals like the salmon fish or any other animal, we can get the products of anti-cancer products in this fish or the other animal into which we have incorporated this particular gene. So in this way, bioprospecting field involves in the identification of new genes which are useful for our use, or which are useful to cure our diseases, which are useful to uh, meet the demands of our food requirements, all those things. So all these things are coming under this aquatic biotechnology. And the important one is medical biotechnology. So this medical biotechnology involves in the whole spectrum of human medicine. All types of medicines, all types of diagnostic techniques will be developed by using this biomedical biotechnology field. So preventive medicine will be developed, diagnostic tools will be developed for many diseases, and treatment for human diseases. So drugs are also, curing drugs are also will be made or produced by this medical biotechnology knowledge. And new information from human genome project also, already the human genome project almost already finished. So this knowledge of human genome project is being utilized in gene therapy practices and stem cell technologies. And what are the pros, what are the benefits of this biotechnology? Let us see. So this biotechnology can improve the health and reduce hunger both at the same time, and it creates flexibility within the food chain. So we need not to kill other animals indiscriminately, as we are able to increase the production, so that we need not to kill the other animals, so that we can conserve the food chain and food web. It allows us to preserve resources, so resource preservation will be made possible. And it helps us to minimize waste products. So wasting the products also can be minimized, and it can reduce infectious disease rates. It also offers us medical advancement opportunities. So all these are the benefits of biotechnology. So apart from benefits, some negative points are also there in this with this biotechnology. It creates an all or nothing approach. So, for example, we are doing some research in this biotechnology field. So either it will be successful or it will not be useful at all. So all or nothing approach. So a partial development will not be seen. So either it is all success or no success at all. So if it is no success, if the research is no success, then we would have to lose a lot of money. Why? Because all these biotechnological research activities require a lot of funding. So that fund will be wasted. And it is a field of research with many unknowns. So as it is newly developing branch, so many unknowns are there. many things are not known to us, so that a lot of problems have to be faced by the researchers. It can be used for destruction. So the knowledge of this biotechnology also can be used for destruction, as we are hearing the many rumors that China country has designed this COVID-19 vaccine, COVID-19 virus, to destroy other nations. So if that is true, that would be the best examples for this point. So that is practically possible. So if this technology falls in the hands of bad people, they can use this for the destruction of their enemies, their enemy countries, like that. So it could ruin croplands also. So we know all types of all angles of this biotechnology. So scientists may think that this would be good, so that let us produce this type of genetically modified organism, like that. But they don't know the consequences of that genetically modified organism. So it may ruin the croplands, or it may ruin the other animals which are present in the ecosystem. So that is also possible. Why? Because many unknowns are there in this biotechnology, so that consequences cannot be uh, guessed, or consequences cannot be mm, foreseen by the scientists. So this is also another uh, con of this biotechnology. So with this, today's topic is over. Let us meet in next class with another topic.